I'd like to introduce two in-person presenters today for this first presentation of this session. The first is Dr. Kara Nelson, who is the Bloom Chancellor's Chair in Development Engineering and a professor of Civil and Environmental Engineering. In her research program, Professor Nelson aims to develop innovative technologies and accelerate the adoption of environmentally sustainable and socially equitable water infrastructure and practices around the globe. And Camille Brule is a second-year PhD student in Kara's lab who is interested in research that focuses on closing the sanitation equity gap in developing contexts, specifically in regard to nutrient recovery from urine and feces. Dr. Kara Nelson and Camille Brule will be presenting an introduction and evaluation of ammonium recovery from urine using ion exchange. Come on up.
And just a reminder to folks up here, if you can speak up so folks in the back can hear you.
Thank you.
Yeah. All right. Good morning, everyone. And afternoon or evening for people on Zoom if you're in other parts of the world. So what an honor to be kicking off the third day. So I'm going to be starting the presentation and trying to frame where ion exchange might fit in the innovation ecosystem for urine technologies. And I'll touch briefly on some work done by Utsav Shashwat, who was a postdoc in my laboratory until very recently. And then I will hand things over to Camille to share some of the work that she's been doing and thinking about for her PhD. So first of all, how does ion exchange work? So for targeting nitrogen in the urine, we target the ammonium ion. So we are planning for this technology to be applied after the urine has been hydrolyzed. And so we've got NH4 plus present in the urine. And we use a weak cation exchange resin, which has these negatively charged carboxylic acid groups. And so because ammonium is positively charged, when urine passes through a fixed bed of this resin, it will attach through electrostatic bonding to the negatively charged sites. And we can target ammonium that way. We could also target phosphorus, phosphate. In some of our previous work, we have demonstrated that we can also remove phosphate through using an anion exchange resin, or we can precede the cation exchange column with struvite precipitation. And I've heard there's some Will Tarpe fans here. And so this is work that was started with Will Tarpe when he was a PhD student at UC Berkeley. At the end of this process, what you have is the liquid urine with very little nutrients in it. I also wanted to tie in this process with the use of biochar, because we've heard a lot about biochar the last few days. So biochar can also be used as an adsorbent. The mechanisms that the nutrients attach to biochar through are more complicated than with ion exchange, but it's very similar. In the case of biochar, if you're using that as the adsorbent, then you apply the nutrient-enriched biochar directly to the land. But in the case of ion exchange, we want to elute the nutrients off of the resin, so we can reuse the resin. And so there's a second step where we pass an eluent, and most of our work in the past has been based on sulfuric acid, and we pass the eluent through the column, and then we produce a high-strength fertilizer, in this case, ammonium sulfate. So what's unique about this process is that you can separate these two steps, both in space and time. So the nutrient capture, and I'll continue to focus on nitrogen for the rest of the talk, but remember that this can also be used for phosphate, and we also get some potassium with the nitrogen. So you can separate the capture step from the recovery step. And so this allows it to be downscaled and potentially done without needing any energy. So like the adsorption could occur with these cartridges installed right at the toilet. And then the cartridges are what gets transported, and you can achieve, we think, about a five-fold volume reduction. And then so the fertilizer production step can be done at a more centralized site, and then the residual urine management can adapt it to the context. So we've been working on trying to think about what scale up of this process would look like in two very different contexts. The first one is San Francisco, so representative of a high income, high density urban area. And we, a few years ago, did a life cycle assessment where we modeled a scenario where 50% of the toilets were urine separating and outfitted with these ion exchange cartridges. And the residual urine was sent to the local wastewater treatment plant. And then the cartridges were serviced by a service provider, kind of like a recycling truck that would do routes and collect the full cartridges and replace them with empty cartridges, and then transport the cartridges to a centralized facility to regenerate them and make the fertilizer. So in our LCA, our baseline scenario was achieving the same level of nitrogen removal at the centralized wastewater treatment plant using nitrification, denitrification. And we were able to demonstrate that this urine source separating scheme ended up with significantly lower greenhouse gas emissions than the centralized treatment option. But something that surprised us was that the largest contributor to the greenhouse gas emissions was not the trucked transport, but the use of sulfuric acid for eluting the ammonium off of the columns. And so a lot of our work since then has been focusing on trying to make that part of the step or the process more efficient. And I'll speak about that a little bit in a moment. The other context that we've been working on, if you were listening yesterday, I brought this up during the container-based sanitation panel because we work with Sanergy, who's a sanitation provider in really high density slums in a very low income region in neighborhoods in Nairobi. And they do source separation with dry toilets and then collect the waste in separate containers. Right now, they service about 30,000 users use their toilets every day. They collect all the urine, and the urine collection is one of their significant operating expenses, and they don't do anything valuable with it because they haven't found a cost-effective option. So what we are exploring is whether we could replace this container where the urine is stored with these cartridges. And then they would be hauling cartridges, again, hoping for that 5-fold reduction in the amount of volume that they have to haul. And then preparing the urine for on-site disposal through infiltration in the soil. And the challenge is just that there's a very small space. So often the only space available is the space directly under these toilets. Okay, so back to our work in the lab. As I said, we've been focusing on trying to make this elution step more efficient. And so we are interested in having that part of the process result in lower embedded greenhouse gas emissions, improve stoichiometric efficiency, which is just, if you imagine in the elution, there's a counter ion that's kicking off the ammonium. So we would love for that to be a 1-to-1 ratio, which would be like the highest efficiency use of the eluent you could have. And then we'd love for this fertilizer to be as concentrated as possible. So some of the work that Utsav did, it was almost all conducted in the lab with these small scale, bench scale columns. So you can see them packed with resin there. And one of the things he compared was using different eluents or mixing the eluent with other possible chemicals that could serve the same purpose. So I'll just highlight this example here. We found that you could replace half of the sulfuric acid with sodium sulfate and achieve similar stoichiometric efficiency and concentration factor, but it would be lower cost and have lower greenhouse gas emissions. So that's all I'm going to share on the really technical side, but if you want to learn more, I'm happy to talk about it. And we're also preparing a manuscript with a whole bunch of other results. And with that, I will turn things over to Camille.
So if you were at the summit last year, you might have seen the virtual tour of our ion exchange reactor. It's since been scaled up from that bench scale system. I clipped together some footage of what it looked like 12 months ago versus now. This has been the focus of my PhD so far. We finished all of the hardware components, and now the reactor runs with 34 different solenoid valves, 4 peristaltic pumps, and it uses a combination of Arduino and Raspberry Pi. We have not put real urine through it yet because I'm still working on some leaks, but we've been operating with synthetic urine and have successfully run it 3 times so far. And so the next step, hopefully, is real urine soon. But as you can see, there's been a bit of aesthetic, but also functionality improvement in the last 12 months. And okay, so future works that I'm going to focus on for my PhD, like Cara talked about, focusing on the elution step. We're putting neat sulfuric acid through a column that's fully saturated with urine, or ammonium is attached to the beads. And Cara was saying we want a high concentrate fertilizer to come out of there, but right now we're getting ammonium sulfate, but there's still some sulfuric acid in that. So I'm curious if you can just recycle that eluent back through another column until it gets more concentrated with ammonium sulfate, and if that could be a tactic to reducing our reliance on sulfuric acid just by recycling the eluent more. I'm also curious what happens to the resin themselves in the column after being regenerated multiple times. The manufacturer doesn't publish the lifetime of these resins, and that's a really big determinant factor of the cost effectiveness of ion exchange for its feasibility of implementation. So if you take a closer look at the actual beads in the column, they're undergoing this constant cycle of being deprotonated and having an ammonium attached to it, and then having the proton from sulfuric acid or whatever eluent you use, reprotonating it. And I'm curious, over 10 cycles, 50 cycles, 100 cycles, what happens to the resins if the micropores get blocked, if the functional groups get harmed. So that's the other direction I want to focus with my PhD. And then future works that I don't have any specific experiments planned for, but would love to investigate, would be looking at the microbial activity in the actual fertilizer that is produced. Because we're using such a strong acid, it's at a really low pH. So I would hypothesize that there's not going to be a whole lot of microbial activity, but actually looking into that and proving that would be really important for the actual implementation of this. I'm also curious about how biological nitrification, what's occurring on the adsorbents themselves, and taking that into consideration with the nitrogen balance on the entire ion exchange reactor. So we just wanted to acknowledge some other researchers and collaborators we've worked with. Cara is a really big fan of this sign in Berkeley, so we put that in the presentation.
Oh.
For those on Zoom, it usually says speed checked by radar, but someone vandalized it in a really great way. So thank you guys for listening to our presentation. If you have any questions about the ion exchange reactor or details of how we make anything run, please talk to Cara and I later.
Wonderful. Thank you both. That's great. I'm very curious to hear what happens next on that. Our next presentation is via Zoom. Jeff Meilander will be presenting, and Jeff is a biology PhD candidate at the Caparaso lab in Northern Arizona University, where he studies the microbes of human fecal composting using composting toilets. He'll be presenting microbial succession of human feces in self-contained composting toilets. And we can see your screen quite well. Great. Is this recording or yeah? Okay. I'll stop talking to Jeff and I'll turn this up.
I'm a P. Hi everyone, my name is Jeff Meilander and I'm a PhD candidate in the Caparaso lab at North. Hi everyone, my name is Jeff Meilander and I'm a PhD candidate in the Caparaso lab at Northern Arizona University located in Flagstaff, Arizona. I'm excited to share part of my doctoral research titled The Gut to Soil Axis, microbial succession in human excrement composting. So let's begin by lifting the lid on what we found in our composting toilet investigations. I'll start with a very brief overview of the methods. 11 participants used different composting toilet systems, as you can see here in the image. The Sunmar Excel non-electric, the Nature's Head, a 3-barrel system, which for those of you who attended the recent EcoSan permitting group may recall learning about this system from Catlow, and a simple homemade system with a 19-liter bucket. The participants used the toilets for 3 weeks, diverting urine before each use. They mailed us 6 self-collected samples, which included 3 weekly human excrement samples, 1 swab from inside the collection chamber, 1 of the bulking material, and 1 from the floor around the toilet. After 3 weeks of use, I transferred the material into 19-liter buckets, and they were stored in a climate-controlled greenhouse between 13 and 16 degrees Celsius. Each week, the material was aerated and homogenized, and 2-gram samples were collected for a total of 52 weeks. The compost remained in the buckets until week 133 before being added to an actively managed thermophilic compost pile at a sustainability education center I started. The DNA from these samples was extracted, and we sequenced the V4 region of the 16S rRNA gene to identify the bacteria. This is a process by which we can get an approximate relative abundances at the genus level of the bacteria present in each sample, and therefore gives us a high-level overview of the composition and dynamics of the microbial communities. This specific technique, however, doesn't give us sufficient resolution to identify pathogens. Instead, we used quantitative PCR to track potential pathogens, including E. coli and C. perfringens, as well as culturing E. coli. We also ran assays to identify bacterial load in enteroviruses, sequence for protists, and we're now planning to sequence for fungal communities and conduct shotgun metagenomics to identify the genes of all organisms in our samples. Today, I'm just going to focus on the high-level bacterial community compositions. Over 1,000 samples represented by these dots are plotted in this principal coordinates analysis of the unweighted unifrac distances, which represents the similarity and dissimilarity between samples. The closer 2 dots are together, the more similar their microbial composition is, and the farther apart the dots are, the more dissimilar they are. To help you better understand these data, let me quickly orient you to this figure. You can see that the human excrement samples here in brown form a tight cluster, representing more similar microbial composition, which is to be expected. The human gut is a unique microbial habitat, so you tend to see similar organisms living there across different individuals. The bulking materials used by each participant in the composting toilets are in green. Reference samples, which consisted of publicly available soil samples, are black, and previously collected food and landscape compost samples are red. These samples form a more diffuse cluster in the middle. And finally, the big smile in gray are the weekly human excrement compost samples from each bucket. Now this plot shows the same data, but the samples are color-coded by week. And you can see there is a clear progression from the early time points to the later time points, showing that the microbiomes are becoming more dissimilar from the human excrement over time along axis 1 at the bottom. Given this pattern, we wanted to track and show the microbial succession over time for each bucket. Again, these data points are the same data you previously saw, but the data points for the specific buckets, in this case bucket 1, are highlighted in black. Time point 1 is labeled as start. And the large distance between time point 1 and the participant's human excrement represents a rapid initial transition, which is then followed by less variability in consecutive time points. Time point 52 is labeled as end. Consecutive time points are then connected by a black line and illustrate the trajectory of succession over time. For comparison, The blue stars and line show the average trajectory of all of the buckets. The dotted lines connect the participants' excrement sample inputs along with the bulking material inputs to the start of the sampling period. And by time point 52, bucket 1's trajectory aligned with the average and was significantly different from the original human excrement. The microbiome at time point 1 in bucket 4 showed the fastest transition away from human excrement compared to all buckets. It also aligned closely with the reference samples. Manufacturer guidelines recommend aerating the compost every 2 weeks using a compost crank, starting with the older barrels to prevent the introduction of pathogens from the newer material into the more mature compost. However, in doing so, this likely inoculates the newer material with microbial communities representative of finished compost and therefore influences the successional pattern you see. Bucket 5 had foul odors throughout the study, which was probably due to improper use or poor airflow inside of the bucket. The pH was initially 10 and moisture was nearly 100% for the whole experiment. Interestingly, despite starting close to human excrement, bucket 5 showed the largest extent of change and ended up closely resembling the microbiomes of reference samples and bucket 4. Finally, bucket 7 started to transition away from human excrement, but stalls after time point 4, remaining closer to human excrement than average. However, bucket 6, which isn't shown here, belonged to this participant's spouse and showed a similar stalling pattern, but something caused it to quickly progress at time point 31, ending here at the yellow smiley face. Bringing all 4 buckets together shows significant differences in the extent, rates, and trajectories of microbial succession over time. And just to point out, these 4 buckets displayed the most prominent changes of all the buckets, but each bucket has its own unique characteristics. Other interesting observations happened when we analyzed taxonomic bar plots. In this figure, each bar represents one time point, and each color within the bar represents a specific taxon. Buckets 2 and 3 were created by spouses, and both displayed the spontaneous appearance of Arachidocockus and Rhodanobacter at time point 22, represented by the dotted black line. This pattern was unique to these buckets and helps us identify specific organisms and time points to investigate more closely. Overall, microbial succession in human excrement compost shifted from a fecal microbiome to one resembling soil and traditional compost. Later stage taxa were involved in nitrogen cycling, humification, and lignocellulose degradation. Additionally, key taxa identified were associated with plant growth promotion, bioremediation, production of iron chelating compounds, and antifungal activity. This analysis raises several important questions, such as how can we manage composting toilets so their microbial succession follows a trajectory similar to bucket 4 or fixes a stalled transition seen in bucket 7? Can these trajectories be controlled through inoculation? What are the active genes and enzymes that occur during these changes? Were abundant or rare taxa more influential in driving these changes? And what microbial interactions occur that may lead to these patterns? This provides a useful baseline for comparing future studies and guiding further research. Understanding these dynamics can enhance composting efficiency, help identify indicators of compost readiness, and provide insights into reducing pathogens, pharmaceuticals, and antibiotic resistance contamination. In order to answer these questions and accelerate composting research, we're developing an interdisciplinary composting microbiome living lab on NAU's campus. Because of our close proximity to the Grand Canyon, we will be using excrement from river trips collected in watertight containers called groovers. We're planning to start at Site 1 and expanding to Site 2 if needed. Both sites are near NAU's food composting site, which we hope to better manage and study for future co-composting experiments. Our proposed experiments include mesophilic composting in larger bins, as well as thermophilic composting. The results from both experiments can be compared to each other, but also to the data set I just shared with you. We also plan to run spike-in experiments where we add known concentrations of pathogens or pharmaceuticals to monitor their degradation and the associated microbial changes. And last, we will conduct similar vermicomposting experiments using worms. Collectively, these experiments will help optimize composting, identify bioindicators, and explore useful bioinoculants. Thank you for your time, and please reach out if you're interested in collaborating on a project or learning more about our research. Enjoy the summit.
Right. Well, thank you. Thank you to Jeff. Jeff, who is not here today, that was a recording. I saw some, at least one question going up in the chat, which is great. I think we can probably get questions to Jeff. So if anyone does have questions, please do continue to put those in the chat. And I forgot to say at the beginning, please put questions to anyone in the chat. In addition, to Kara and Camille, if you had questions for them, please pop those in the chat. They'll either answer them in real time or we'll circle back at the end for Q&A if we have some time at the end for the panelists to come back up. The next presentation is from Nebiyat Nigusie, a PhD student in Addis Ababa University, Ethiopia, who is studying in Dr. Ade Felike's wastewater laboratory. Nebiyat is presenting method validation, detection, and quantification during assessing fate of selected antimicrobials through struvite making from source-separated human urine by liquid chromatography-tandem mass spectrometry, excuse me. And Julia, is that a recording as well? Right, so we'll just pause a moment while Julia gets that onto the screen.
Nebiyat Nigusie from Addis Ababa University, Microbial, Cellular and Molecular Department. I am going to present a work about method validation, detection and quantification of selected antimicrobials from struvite using ultra-high liquid chromatography, tandem mass spectrometry. The already identified facts about struvite will be introduced, then the methods of spectroscopy will be explained. This is followed by revealing the results and discussion and will be concluded with concluding remarks and recommendations. At last, organizations and people who support this work will be acknowledged. We prepared an eco-friendly fertilizer called struvite from human urine, though there are concerns of micropollutants arising from human consumption. These micropollutants are, for example, antibiotics. We assessed the presence of antibiotics which are prescribed commonly in Addis Ababa. The assessed antibiotics were ciprofloxacin, cefotaxime, oxytetracycline, tetracycline, and doxycycline. To detect and quantify antibiotics in struvite, fresh urine, and stored urine, an analytical method called ultra-high liquid chromatography-tandem mass spectrometry was used. To use this method to detect and quantify antimicrobials, a method validation was done. The U.S. Environmental Protection Agency standard for 2007 was followed in order to test the specificity, accuracy, linearity, and precision of the method utilizing the data obtained from spiked standards. When we go to the methods, fresh urine sample was collected from Addis Ababa town called Piazza. In 1 hour of collection, the sample was transferred to bioinstrumentation laboratory, Addis Ababa University, for storage to sanitization to occur. After storing for 20 days in sealed jerrycans, struvite was produced after adjusting the urine pH to 10 by sodium hydroxide solution, followed by adding equimolecular amount of magnesium chloride to phosphate. Thereafter, both the prepared struvite, fresh urine, stored urine, standard fortified non-history urine were passed through solid phase extraction, followed by measuring method validation and quantification of residual antibiotics. This table is an illustration of mobile phase gradient used, which was helpful for separation of multi-microbial mix in the samples according to chemical nature of the antibiotics. The mobile phase A is water and 0.1% formic acid. The mobile phase B was prepared using acetonitrile and 0.1% formic acid. Mobile phase C was prepared by mixing methanol and 0.1% formic acid. We have conducted the experiment aiming in search of 7 commonly prescribed antibiotics in Addis Ababa city, so we had succeeded in validation of the method and quantification only for 5 of them. Here is a displayed chromatogram for standards fortified in history known urine for all the 7 antimicrobials explained. The slide displays the chromatogram of the 4 antimicrobials identified in fresh urine. And this is the slide shows for the 4 antimicrobials traced in stored urine. This is the result of the method validation. The targeted antimicrobial specificity shows no interfering peaks at the retention time. Calibration curves for the linearity measure was produced, and it was discovered that the linearity coefficient fell between 0.9945 and 0.9778. The mean recovery percentage varied for doxycycline from 88.51 plus or minus 8.15 to 92.86 plus or minus 7.26 for tetracycline. On the other hand, the intraday precision met the Environmental Protection Agency criteria, ranging from 5.05% for oxytetracycline to 12.59% for ciprofloxacin. Conversely, the inter-day accuracy varied between 16.7 and 8.1 for cefotaxime and ciprofloxacin respectively. The limit of detection and limit of quantification values for candidate antibiotics ranged between 0.91 to 2.93 microgram per kilogram and 3.03 to 9.77 microgram per kilogram respectively. According to the above validated method, we quantified 4 of the antibiotics in search. Here is the result found in both fresh urine and stored urine, but none was detected in struvite. This could be the struvite processing pH or microbial degradation at the cause during struvite drying. These reasons are supported by former studies. Conclusion and recommendation: the struvite was found free from selected microbials. Both fresh and stored urine harbored antibiotics. In recommendation, struvite making process or drying process may affect the stored urine persisted microbials. Therefore, the effect of these processes should be studied in relation to microbial degradation in struvite. We would like to acknowledge the people who support and the organizations and the people who support this work. First, we would love to acknowledge the Rich Earth for giving this chance. Then we would love to acknowledge Addis Ababa University Thematic Fund and USAID Peer Fund and all the people who contributed in this work. Then we would love to thank you for your attention. If you have any question or suggestion and comment, you are welcome. To find more about this work, it's under review and it's released on a pre-review database.
Thanks. Really good, thank you. So if anyone has questions for Nebiyat, you can put those in the chat as well. The next question, or the next presentation rather, is from Maano Tshimange, a second-year PhD student in chemical engineering and processing at the University of Surrey in the UK. Her work focuses on membrane fouling and nutrient recovery from urine. Maano is presenting evaluation of membrane fouling during dewatering and nutrients concentration from fresh urine by forward osmosis.
Hello, can you see the screen?
Yes, we can.
All right. Good afternoon, everyone. My name is Maano. So I'm going to give you a brief presentation about my current work at the moment on evaluation of membrane fouling during dewatering and nutrient concentration from fresh urine by forward osmosis membrane. So I won't delve down to the introduction about urine and urine source separation because that has been massively done. As everybody knows, there are so many technologies at the moment that are being used or even being explored to recover nutrients and water from urine. So one of the technologies include a membrane filtration system, such as reverse osmosis and nanofiltration. However, the problem with this system is that it requires a lot of energy, and also it is prone to fouling. So there's an alternative membrane system known as forward osmosis membrane. So with forward osmosis membrane, it uses or rely on osmotic pressure gradient to extract water. In this case, it will be urine, while at the same time concentrating and rejecting nutrients. So nutrients such as phosphorus can further be processed to produce struvite fertilizer. And then also the diluted raw solution, in this case, I'll give an example. For example, like NaCl can be further processed to produce clean water. So the advantage of FO membrane is that it has low fouling tendency and also consume low energy as compared to the traditional membrane systems. Although it is less prone to fouling, it's still subjected to fouling when it treats a lot of really feed water system. Feed water. That affects the membrane performance and also reduces the membrane lifespan. So as we all know that urine contains a lot of different compounds, including pathogens, organics and inorganics. It obviously, when we treat the urine, the membrane fouling is going to occur. However, there hasn't been a lot of studies that is being done focusing on membrane fouling while recovering resources from urine, hence why the purpose of my study. So at the moment so far, we have worked only on synthetic urine. So we use synthetic urine that contained organic and inorganic compounds that are dominantly found in fresh urine. So all the work was done on a bench scale mode. Basically, what happened was that we did that in a multi-cycle until 80% of the water was recovered. So this was done in 3 cycles, and then after the fouling test, we clean our membrane to check the extent of fouling and also the efficiency of our cleaning method. And then once that was done, then we further took our membrane for membrane characterization, as well as also the fouled membrane. We took it for membrane characterization. We also use different analytical methods to analyze the concentration of the concentrated urine. So from the results that we got, basically this was done under two cross-flow velocities. The results were quite similar, really, and we found out that the flux decline was mainly due to the dilution of the water, which reduces the driving force of extracting water from urine rather than membrane fouling. So this was also supported by our results when we compared the baseline flux with the flux that we the flux of fouled membrane. And then after cleaning, we managed to recover 100%. So meaning that the fouling was reversible, and also our physical method that we use for cleaning was effective. So in terms of the solute rejection, solute rejection were 100% for most of the solutes except potassium. Except for potassium, as it has a high permeability due to a small hydrated radius, which prevented the high rejection. So this is the SEM images of our fouled membrane. As you can see, there were precipitants on the surface of the membrane. However, when you clean the membrane, it was more or less similar to the virgin membrane. So again, the fouling was reversible, and it was really not that significant. In terms of EDX analysis, we identified magnesium, phosphorus as the main or the dominant foulants, followed by potassium and a bit of sodium and chloride. However, again, when we clean our membrane, there was really low elements, and then as you can see, when we compared with the pristine membrane and also the clean membrane. Then we use FTIR just to see whether there was any organic foulants, and from the peaks here of the pristine membrane and the foul membrane, they were quite similar. So we concluded that there wasn't any organic fouling that occurred. And then we use AFM to check the roughness of the membrane, to check whether or not the foulants had any impact on the roughness of the membrane. So then we noticed that the roughness increased from 3.6 to 6.32. And then, but then decrease again after cleaning. So again, the fouling, even though there was a bit of roughness, but it wasn't, that severe, because also when we clean, it went back to the same roughness level as the pristine membrane. Similar results in terms of the presence of the foulants on the membrane surface cause lower contact angle values, indicating that the fouled membrane become more hydrophilic. However, when we clean it, the contact angle was more or less the same as the pristine membrane. So just to conclude, we didn't really found any significant fouling with our synthetic urine, and that the water flux decline was due to the dilution of our draw solution. So because of that, at the moment, we are busy working on real urine as we believe that we're going to find more severe fouling and the results will be different compared to the synthetic one that we use. Thank you so much.
Wonderful. Thank you very much. And any questions, please place those in the chat. Our last presenter today is Dr. Charles Buregeya Nyiwagaba, the founder and executive director of Sustainable Sanitation and Water Renewal Systems, a local NGO working in Uganda, and also is an associate professor of civil and environmental engineering at Makerere University. Dr. Charles Buregeya Nyiwagaba is presenting circular fertilizer production by solar thermal-driven passive drying of acid and base-stabilized fresh human urine.
Hello.
Hello. We hear you very well.
Yes. Okay. Very good. Is the presentation going to load from that side? I had sent it, or should I load it from here?
If you can load it from your side, that's great. And if not, let us know and we can get it pulled up from our end.
Okay. Okay. Let me try that and see. Share. Okay.
Good. We see that.
You can see it?
Yes, we can.
Okay.
It looks great.
Okay. Okay. Thank you very much. So, thank you very much. I'm going to make a presentation on the circular fertilizer production by solar thermal-driven passive drying of acid and base-stabilized fresh human urine. And I've been introduced, so I don't go through the details again. So I'll give a small background, and then I will talk about what we did and what were some results, conclusions, way forward, and ongoing work. Let's say this background has been presented all along, but we know that human urine is really a primary source of plant nutrients. And therefore, if we recycle it, we will be moving towards achieving sustainable food production, as a human being will always produce urine for as long as they live. However, we know that urine is bulk, and approximately 95% of it is water. And therefore, if we have to apply given a quantity, for instance, 90 kilograms per hectare, we need about 15,000 kilograms of urine. And yet, if you are looking at some kind of synthetic fertilizer, you could apply only about 200 kilograms to achieve the same fertilizing rate of application of ammonia, of nitrogen. Therefore, that bulkiness of the urine is in fact a problem for two major reasons. So it will lead to costly transportation, and it will lead also to costly application. The fact that where the concentrations of people are will be, let's say, urban areas or some institutions, and where the farmland might be, could be some distance away needing transportation. Therefore, but also the liquid urine normally smells, and that also could cause social cultural issues or acceptability issues. And therefore, we need to reduce the bulkiness and produce a concentrated fertilizer that will not smell or smell less than if it is liquid urine. So we set out to reduce the bulkiness of the urine, and we did this by concentrating the fertilizer in the urine into a solid product. We started by stabilizing urine by knowing the fact that the urea that is contributing to the nitrogen in the urine at excretion will immediately convert to ammonia in a reaction catalyzed by the enzyme urease. So we created conditions under which this enzyme urease will not act by alkalizing urine using wood ash, and also by using hydrated lime at different concentrations of 100 grams per liter for wood ash and of 15 grams per liter for hydrated lime. We also tried acidification, whereby we achieved, we dosed 5 grams per liter of citric acid to produce a citric acid stabilized urine with a pH less than 4. These conditions of pH over 11 and pH less than 4 were considered to degrade the degradation process that happens by converting urea into ammonia using urease catalyzed reaction. So what we did was to start to understand what would happen in a small solar concentrator and see what kind of temperatures that you can realize. And in this, we found that, first of all, we looked at the irradiance and we obtained this happening between 750 to 800 and there was occurring between 14 hours and 17 hours. This kind of trend was also followed by the temperature that actually was increasing around those times, and also with the moisture decreasing at the time when humidity decreasing at the time when the temperature was highest. We subjected samples of 1 gram to isothermal tests conducted 1 gram of samples at 50 degrees Celsius with a flow of 1 meter per second in a gravimetric analyzer. And then we found some interesting results whereby the urine was was stabilized by ash dried faster, of course, also because of the fact of the absorption of the urine by a lot of ash that was applied, and then calcium hydroxide followed, and the stabilized urine by citric acid took longest to dry because very little had been applied to achieve the pH required. What we did was to conduct some kind of modeling and subjected the results to 10 models for both ash stabilized urine, lime stabilized urine, as well as citric acid stabilized urine. And we conducted and performed statistical analysis from these models and found that the two-term model was best predicting the drying for wood stabilized ash. Wood ash stabilized urine, and whereas the page model was best describing the drying for lime stabilized urine as well as citric acid stabilized urine. The 10 models that were run included the Newton, the Page, the Henderson, the logarithmic, the Wang and Singh, the diffusion, the Varma, the two-term exponential model, the Midili, and the those were the models that were followed. Now, what happened was that we found that the wood ash and calcium hydroxide, as well as citric acid, at dosages of 100 grams per liter, 15 grams per liter, and 5 grams per liter were sufficient to give a pH that could block hydrolysis. And the stabilized urine attained pH greater than 11 for ash and calcium hydroxide stabilized urine, and pH less than 4 for citric acid stabilized urine, the conditions that were responsible for blocking urea hydrolysis. The two-term model was formed to best suit the drying curves of wood ash stabilized urine, whereas the Page model predicted the drying for calcium hydroxide stabilized urine and citric acid stabilized urine. And the drying method that we used, when after checking these conditions in a small solar concentrator, as well as some gravimetric analysis, we subjected our samples to drying on the sun. And then we found a raw nitrogen, nitrogen to be 2.1 to 2.7, which was lower than 10% as reported by SIMA in 2021. What happened was therefore that we need to improve our drying in a way that we get values that are closer to those that have been reported in previous literature. However, we didn't end there, but then we transferred this one in the field conditions to try to look at the realities in collecting urine, as well as the realities in drying urine under the field conditions. As you can see there, is a simple modification of a urinal in men's urinals. It's very easy to realize this in men's urinals, of course, difficult to realize this in female urinals. And in that case, maybe one will be looking at urine diversion that has been heavily discussed over the last 2 days. Then what happened, our treated or stabilized urine was exposed on the sun under the tropical conditions we have here in Kampala, whereby sometimes you have a hot day that the temperature goes somewhere between 30 to maybe around 33 degrees Celsius. However, interestingly, that when you have a rainy day and you are drying under such conditions, then maybe you have to cover. And also the rate of drying might be slower than the rate of absorption of carbon dioxide. So the pH might end up reducing before you actually achieve the dryness that you want. So the dried urine that we obtained from that drying was actually applied under greenhouse trials at the Ugandan National Agricultural Research Organization that helped us or supported us to conduct these trials. And we used the ratios that were obtained under the greenhouse conditions that gave the best biomass yield under field conditions. And we did field trials in central Uganda, again, working together with the National Agricultural Research Organization. And from there, we really obtained some interesting results. So that you can see that we had dried urine, we had liquid urine, and then we compared this with the ammonium phosphate. And these were applied at ratios of 0 to 2.0, where X is the recommended rate of DAP application in central Uganda. And we applied this in split plot randomized complete plots, whereby the main factor was fertilizer type and the subplots as the fertilizer rates, which were replicated 3 times. As I said earlier, the dried urine that were used in these experiments, as well as the liquid urine, had these ratios of nitrogen, almost 10 times for nitrogen content in the dried urine as compared to the liquid urine, and other ratios of P and K as shown over there. Interestingly, we found that the maize yields were significantly higher with the DAP at the rate of 5,881 kilogram per hectare as compared to the control. And the dried urine yielded 5,317 kilograms per hectare, whereas the liquid urine yielded 5,309 kilograms per hectare. We didn't find any significant difference in maize yields between the dried urine and the liquid urine. We found that the use of the DAP resulted in 20% increment in grain yield over the control, whereas the dried urine and the liquid urine increased maize yields between 9.1 and 8.9% respectively. Overall, we could see that the maize yields did not significantly differ among the fertilizer rates, and these preliminary findings show the potential of dried urine to increase maize yield, grain yield here in Uganda. However, we need more studies to validate the benefits of dry urine in crop production. What you can see here is basically this small proportion or increases could also have arisen because of the fact that the soil on which we did the tests was probably having some background amount of nutrient content. And therefore, if you deal with the soils that are really barren, I believe that you could register higher yield, higher green yield, looking at the dried urine, also liquid urine. But the importance of dried urine over liquid urine is, as I explained earlier, that the liquid urine would be barochem. We apply quite a lot of it, whereas for the dried urine, we apply very small quantities, since the quantity of fertilizer, especially nitrogen in the dry urine, was almost 10 times higher than you could find in the liquid urine. So thank you, and I'm really happy to share what we have done, and we look forward to doing more and continuing sharing with colleagues here in the future. Thank you very much.
Wonderful, thank you. We're in the peculiar situation now of having all of our speakers presented, and we're not over time yet. Our session goes until 10:20 and it's 10:07. So there's an opportunity for extensive follow-up questions, I would say. Has anyone who presented gotten any questions in the chat that they would like to answer verbally? Or is there anyone who would like to ask a question on Zoom or in person, or have any discussion even? We have a little bit of an unusual moment that we can dig into some things more than just superficially. Yeah, Nancy. Oh, okay. Let's see. We have a thing where we collate these. So, okay, great. So is the one in bold, the yellow, the one that, no, the one. Okay.
But they're worth discussing.
Okay, great. So thank you, Gretchen, for helping with that. It's hard to keep track of what's that, Nancy? Okay.
I do respond, but I can do it in person too.
Yeah, why don't you do that, and then everyone can understand the question.
Well, I actually put a question. So the questions were from Nancy and from Brian Horsley about the pH in the final liquid fertilizer product. And so, which Nancy actually used in a field trial. And so it was, at that time, it was like really low pH, like probably like pH of 1 or something. And so she had to buffer it. And so we have these cool like we follow the pH during the elution cycle. And so you can see like the pH is high. And then as soon as you've eluted most of the ammonium, then it drops, because then the acid's basically running straight through the column. So there's a few options that we're exploring to end up with a product that has not such a low pH. One is you can just stop the elution cycle earlier. And we could, like, if you could automate that, like you could have a pH sensor or maybe a conductivity sensor to help monitor that and know when to stop. But the use of the mixed eluents, like if we replace some of the sulfuric acid with a salt, then we have a little bit more leeway. The pH doesn't drop quite so quickly. And then the experiments that Camille is going to be conducting, like reusing that eluent, that would also have the same effect. Like we basically use up all the H+, and then you would end up with a not so low product. But my question to all of you is, what would be the right pH? So like we think that having an acidic pH is probably beneficial because it would stabilize the ammonia and prevent volatilization and hopefully prevent microbial growth. But we don't want it to be so low pH that people can't directly apply it to soil. So, something that's like comparable to other fertilizer products. So like pH 5, I don't know what to aim for, actually. Anybody have any knowledge to share?
I can speculate. I can use limited knowledge and fast speculation to say.
I love that.
What might matter more is the total acidity more than the pH, because if you're adding it to the soil has a lot of buffering capacity, but the soil also has its own pH that in some cases you would want to be higher or lower.
Yeah.
And so if it's an acidic supplement and it has a substantial amount of actual acidity rather than just being a low pH, then I would speculate that could be good in places where you have alkaline soils. But I'm more used to New England, where it's almost always acid soils. And if you're adding an acid amendment, then you're likely over time going to have to add lime, which is not terribly expensive, but it's like,
I think, but if it's like, so I think if it's still an ammonium based product, doesn't have a lot of total acidity because you have the ammonia buffering. I'll have to think about it more.
Yeah. I think that if you add straight ammonia and then that is nitrified and then taken up by plants, I read this from like soil science extension stuff. I don't have personal experience with it, but what they say is if the ammonia is added as ammonium, and it's taken up by plants as nitrate, then there's no net effect on the pH. If it leaches, then it acidifies. But you're adding ammonia that's been neutralized with sulfuric acid. So I think you're adding ammonium and you're adding sulfuric acid at the same time. So that may overall be
but in the form of, but the sulfate is like a base. Never have acid-base conversations in front of an audience. Like terrifying.
Good idea. Let's stop right there.
What do you think, Nancy? Yeah, we did cost estimates both for the San Francisco, like model scenario and also for the Kenyan scenario. But I can't remember off the top of my head. I know it for Nairobi, we compared what we thought it would take to produce the fertilizer to the current cost of synthetic fertilizer, and you know, if it could be just like under or cost competitive with the cost of synthetic fertilizer, which is like highly subsidized. But that doesn't answer the question directly about what the household cost would be. I will look it up and I'll put it in the chat. Exactly. Yeah. So in our paper, we assumed it could be regenerated. The resin could be regenerated 100 times, but we don't actually know if that's true.
Yeah, come on up and ask it to the microphone.
So I have a question that I think anyone can answer and maybe we can discuss, but I was just thinking, I know we talked about the freeze concentration going to like 2.5 times to 5.5 times, 6 times concentration. And then I know the resin is also looking at like a 5x concentration of the volume. And I know volume concentration is part of the issue, but then also looking at what are the nutrient contents when we have that volume reduction. So just making sure that we're staying in that same kind of nutrient balance. And so with the sun drying, it looked like there was about a 10-time increase in the concentration of the nitrogen specifically. So I just didn't know, is there like a general consensus on like a target for volume reduction, or are we just going as far as we can go? So I just was curious like what everybody's perspectives on that were, and also maybe how we want to be reporting that out to each other, whether that's in nutrient concentrations or in volume reduction values, because I know part of the question is we're trying to make the movement of the urine easier, right? Reduce that volume, and so that it's easier to transport it. But then we also are looking at what is the quality of the final fertilizer. So I just kind of wanted to like put that to the discussion. So.
And I'm just saying, I think once we get it reduced, the farmer doesn't have to go multiple passes. So that's
Yeah, any responses to that question? Yeah.
Question for the user of the farm. What do they want? And what do they want to sell? I'm sure there's like a couple different sort of like needs there.
Great. And if folks can either come up here or speak up from the audience. I've got the microphone right here.
I was just gonna say, I think that, you know, from things that I've read and looked at, a lot of, at least in the US, liquid fertilizers that farmers use would be much more than like even a 10x urine concentrate in terms of their NPK. So I think that in terms of what farmers and farm equipment is kind of like used to using, we can go a lot farther. Now I don't know if there's going to be other reasons that you know, even if we could go that far, like would there be other complicating factors? But I think in terms of just pure NPK, at least in the US, liquid fertilizers that are like kind of on the market and being used are a lot more. I don't have numbers off the top of my head, but I want to say it's well over 10.
I think I've seen some as low as 10, but that's the low end. Yeah.
Yeah, what the X is versus. Everyone about it, transportation too, right?
Yeah. And that, yeah. Yeah, if that wasn't clear on Zoom with the microphone, the last point was the point of it being economically viable will be really important, the concentration to the point where it can be transported, but that's quite variable then depending on the economic factors that are that are determining the viability. I would add one thought also to the question of how do we express the concentration? Is it concentration factor from the starting point? Is it the nutrient level in the product? And that's something that that came up when Kai and I were preparing slides for the first day, because the different technologies, the VUNAS technologies, the sanitation 360 technology and the Brightwire technology, we all talk about concentration factors, but we have different starting points. With, I think, the VUNA installations, they have some flush water dilution in some, but not all of their toilets. So concentration factor from feedstock to product isn't really meaningful unless you know what was in the starting point. And I think what is most relevant in terms of a farmer would certainly be what's the product, and that may be a more equalizing way to discuss the performances of different technologies. What's the ending point? What do we achieve? And it may be that where you start is also relevant, because if you are removing a lot more water, then probably the energy will be higher for a given endpoint than if you're starting at a certain point. But in terms of saying what a technology is capable of, it's probably more easily comparable if we were to talk about the endpoints. But at the same time, if talking about economics, if we're talking about reducing the volume in order to make something more easy to haul, then maybe it's how much water you're taking out that's more relevant, because that is defining your reduction in hauling costs. So maybe we need to just express both of them all the time. And that would probably be great, actually, as we write papers or as we present, to say what we started at, what we ended at, and what percentage water removal that is. And energy usage, yeah. Yeah. And for at least some of these systems, the amount of energy required for removing the first percentage of water is a lot lower than removing the last percentage of water. It's certainly true with freeze concentration and reverse osmosis, and possibly others as well. I saw it. Yes, go ahead, Christina. Yeah.
Climate change technology or any other technology out there. Since my research recently is on salt content, and we're concentrating the nutrients rather than concentrating salts, and therefore, especially things like in other areas that could be accumulating over time and then causing issues down the road. So curious.
One of the main factors that we saw was tracking in our ion exchange work with using these other elements. And so again, it depends a little bit on how we run it, but we are keeping track of how much, how many in which ions end up in the fertilizer product and also in the liquid discharge, because if you're discharging the liquid urine, individual urine to a sewer, like sewer corrosion is also an issue, or you might be discharging salts to a receiving body, or the water might be recycled. So like all of it matters. And we think that we can optimize the system so that we're not adding extra salt compared to what was in the urine originally, if it was like perfectly optimized. That like it wouldn't be a big increase in salt.
Is that using? Sorry, I missed the very beginning. Is that using the kind of actual uric acid pattern? Sodium.
Yeah, either way. What definitely is a bad idea was the sodium hydroxide. The sodium hydroxide adds salt, adds sodium. So that is not going to be, it's also really expensive. But yeah, either with a sulfuric acid or a mixture of sulfuric acid and sodium sulfate, we just use the sodium to bump off the ammonium and not have
My only other question is this season, I don't really know a lot about chemicals that are available for use and like their expense and all of that kind of stuff. But are there other anions that like other anion salts sulfate salts, the other ions besides sodium that would be available that maybe would have the same effect as sodium.
And let's make out our last question.
Looking into that, like, are there like waste acid products that we could be using, or, you know, like somebody else's waste is our. Like, I don't know what the industrial is.
That was the benefit of the sodium sulfate, was it's a waste stream in a process that I can't remember, but it's written in Microsoft's paper that it would, like, we would be using it as a recyclable from someone else's waste, but that's.
But that really gets to, yeah, the overall goal of like circular economies and
Yeah.
locate these kind of, you know, processes so that we can just. So, yeah.
Cool.
Thank you, presenters in person and on Zoom, and also to all these great audience questions. That's been a wonderful session, and we'll move on to the next spot now.